So signal transduction mainly constituted by the proteins which acts as a protein kinases. So those are basically the enzymes, catalysts, so which are responsible for the phosphorylation of the kinases, other kinases. In a way, it activates the other kinase. So that way it forms the signaling cascade. And thus serine threonine specific protein kinases are the important part of this signaling cascade, and they are versatile. Most of the signaling pathways are mediated by the serine threonine specific protein kinases, which was first discovered and well understood afterwards tyrosine kinases. So we discussed about the classification, structure, and then the reaction of the serine threonine specific protein kinases. And in the classification, different kinases we are discussing. So protein kinase A, protein kinase B, protein kinase C, and protein kinase G. So these are all the different protein kinase subclassification. We have already discussed protein kinase A, then protein kinase B is nothing but AKT, that is PI3, PI3K AKT pathway, right? Last class. And now it is protein kinase C is given in your syllabus. So you don't have protein kinase G. G is CGAMP mediated. Protein kinase A is CAMP dependent protein kinase. So today is about protein kinase C. That is the last topic of your third chapter. So already we discussed in the last class about the AKT signal transduction pathways. So PI3K AKT signal transduction pathways are very important. They are responsible for the cells to survive continuously and for its growth. That's why the growth or proliferation is becoming aberrant in the cells which are going to become cancerous. That's how these pathways, the proteins, which are the products of DNA, nothing but when they get mutated, the DNA, these proteins get mutated, and that is nothing but the cancer. Now you understood what is the mechanism of cancer formation. So basically it is because of the signal transduction pathway aberrations. So PI3K AKT pathway is one of the pathway which is very important because this is responsible for controlling cell proliferation and apoptosis inhibition. So these pathways in that way, I told last class that cancer is nothing but because of the oncogenes which are gaining the function of the mutation, and tumor suppressors which keeps a checkpoint on the cell cycle and they get suppressed. So their activity will be lost. The loss of tumor suppressors and gain of function of the oncogenes. That is nothing but cancer formation. Okay. So in that PI3K is very, very critical that it controls. So this was the pathway we discussed, receptor tyrosine kinases when they get activated. Receptor tyrosine kinases, you know, so those are having autocatalytic activity because of which intrinsic cytosolic domain of this receptors gets phosphorylated. So phosphorylated domains are identified by the PI3K. We discussed that it has got catalytic site and regulatory subunits. So through the regulatory subunit P85, this identifies the phosphorylated cytosolic domain of receptor tyrosine kinase, and this PI3K then responsible for the formation of inositol triphosphate. So this is the important reaction mediated by PI3K, which then responsible for many more downstream effects. So this PIP3 is identified by the different proteins which are having the pH moiety. pH is plectrin homology. So this is first identified by the PDK1, and PIP3 is membrane localized. So all these reactions are taking place near the membrane. So PDK1 is getting activated by means of undergoing phosphorylation, and this phosphorylated PDK1 will again phosphorylate AKT. So this AKT phosphorylated by PDK1, further it has to get phosphorylated by another important protein in the cell, that is mTORC2. This is another form of PI3 kinase which is also responsible for second time phosphorylation. So two phosphorylations of AKT leads to complete activation of the AKT, and that's how PI3K AKT is responsible for the promotion of cell proliferation and prevention of apoptosis. Understood? So this way, this is very, very important. So these are the downstream effects of this AKT, for example. So different cell cycle regulators like P27, P21, then cyclin D1, then transcriptional factors. I think all of you now you have understood that. I said again, I'm repeating, whatever may be the signal transduction pathway, the final activators are the transcription factors. Now, transcription factors, molecular biology, next class we will start. So, transcription factors are the one which will induce the transcription. So, these are having the response elements in the upstream of transcription start site for that particular genes, and that response elements are identified by different transcription factors, then responsible for activation of the transcription and expression of the proteins which have got the role in that particular cellular process. So NF-kappa is one of the important, this is known as guardian of genome. It's a tumor suppressor protein in the cell. Then pro-apoptotic proteins like bad procaspase 9 and this is MDM, okay, not MD, MDM should be there. MDM2 proteins. These are all cell cycle checkpoint proteins. So this way, these all proteins responsible for the physiological effects through PI3K AKT signaling cascade. Understood all of you? So that is all about the PI3K AKT, and these are again the same different effectors of the AKT. If you have the question, this illustration you have to practice. So PI3K, then the complete phosphorylated AKT, which is in an active state, is then responsible for all this. This is the same thing, diagrammatic representation. And all these things you have to by heart because these are all new terminology for you. For the time being, like each protein in depth as you start reading, that much knowledge you will get. So here you see this is forward arrow in this type of illustrations or in signal transduction means activation signal, and blunt arrow means inactivation. Okay, so here you see activations and inactivations finally responsible for controlling cellular physiological responses like apoptosis, cell cycle, glucose metabolism, and cell proliferation. So these are all the final events which are controlled by this PI3K AKT signal transduction pathways. Any questions anybody wanted to ask, you can ask regarding PI3K AKT signaling. So if there are no questions, then next last kinase is protein kinase C. So protein kinase C is also another form of serine threonine specific protein kinase that are regulated by three important molecules which are known as diacylglycerol, this is DAG, then calcium and 4-ball esters, TPA. So these three molecules will control protein kinase C. Tetradecanoyl 4-ball acetate, TPA, this is nothing but 4-ball ester, is a tumor promoter was identified as a specific activator of PKC enzymes. So this is a promoter means it's an oncogene. So when it undergoes mutation, then gain in mutation of this TPA leads to the formation of the cancer carcinogenesis. So this is not protein. Okay, I'm sorry. This is not overexpression, gain in mutation, but this, if it is present in more amount, because of this is lipid metabolism product, and this can lead to cancer formation. This is not a gene. This is not oncogene. So now there are more than 10 different subtypes of protein kinase C have been classified into subfamilies, and this classification mainly based on the structure of N-terminal regulatory domain. This is one criteria based on which protein kinase C have been classified, and another one is based on the cofactor requirement of that particular protein kinase. So now There are three different classes of protein kinase C: classical CPKCs, novel NPKCs, atypical or APKCs. So here these are different variants: alpha 1, beta, alpha, beta, gamma, beta 1, beta 2. So four different. And these are classical one is the one which we are going to discuss further. So these classical PKCs are activated by, as we discussed, calcium, diacylglycerol, and 4-esters. Then novel PKCs, NPKCs are delta, epsilon, this is rho, and this is eta. So these forms of novel PKCs are activated by diacylglycerol and 4-ester, but calcium is not effective. So this way, the classification is based on the cofactors and their structure. Then the third one is atypical PKC, APKCs. So this is zeta, lambda, and tau. So these are all the Greek letters you can follow. So different forms of PKCs identified as zeta, lambda, and tau. And these are calcium and DHA independent. They don't depend. So here three: calcium, diacylglycerol, 4-ester dependent. That is classic. Here DAG and 4-ester dependent. Then the third form, atypical, will not depend on this calcium DAG. Rather, it requires some adapter proteins for their enzymatic activity to get enhanced. Okay, so that is about the different classes. Then there is another way of classification apart from this. That is different cofactor. That classification is based on the localization. So depending on the different localization within the cell also is the basis for the classification of PKCs. So here is example: alpha, lambda, and zeta. These are of different classes if you compare based on the structure, but these are widespread in almost all tissues, whereas other subtypes only occur in specialized tissue. So based on the tissue localization also, these PKCs can be classified. So that is about the classification of protein kinases, C forms, and for 2 marks or 3 marks if you get, this is the answer that you are expected to. Then next one is about the structure of protein kinase C. What is the structure? The members of PKC family composed of polypeptide chain with a molecular weight of about 68 to 83 kilodalton. And it has got N-terminal regulatory C1 and C2. Regulatory C1, C2. So it's not catalytic, it is regulatory domain here. That is towards N-terminal. So whenever you write protein polypeptide, it starts from N-terminal and ends with C-terminal, right? N to C. In case of DNA, 5' to 3'. So here, the first N-terminal regulatory domain is C1 and C2. And C-terminal domain can be differentiated in the primary structure of the conventional PKC. So the C1, the conserved domain, is present in the classical and novel. So this we have to understand here. So this is the way. So this is N-terminal and this is C-terminal. So the regulatory domain is towards N-terminal and catalytic domain towards the C-terminal. So towards N-terminal, you have the C1 and C2. Okay. And in the catalytic domain, there is a kinase domain. And these are different forms of the PKCs. Classical PKCs, then this is the NPKCs, normal and atypical PKCs. So here, the C1, the conserved domain, is present in the classical and novel. This is novel and this is classical. Okay, novel means unique, not normal, it is novel. So classical and novel PKCs. And these forms already we discussed. This you need to practice. So in these two, you have C1 and C2 in the structure. So the C1 domain, not C2. So C1 is the conserved domain present in the classical and novel PKC isoenzymes. And this contain cysteine-rich motifs, also known as C1a, C1b elements. So here you can see C1A, C1B here in this depiction is not there, but the C1C here, maybe the CR refers to C1C, C1A, C1B. These are cysteine rich sequences. These are also known as C1A and C1B elements, each with two bound zinc 2 plus ions that mediate. So this region, this C1, C2, C1A, C1 is again divided into two different regions, which are cysteine rich region C1A, C1B. And these regions are the sites to which this diacylglycerol and 4-ball esters are going to bind. So there is no calcium binding C here. 3, right, in the classical. Let's see where the calcium will bind. So atypical PKCs contain C1-like domain with only single cysteine-rich motif that is unable to bind diacylglycerol and 4-ball esters. Atypical PKCs. So these are atypical PKCs. Here you don't have, it has got C1-like domain. It is not C1 domain. C1 like domain is there, one CR is there in atypical PKCs with only single cysteine rich motif which cannot bind with diacylglycerol and 4-ball esters. So based on the activation only this classified, right? Now you are getting no. So first one is classical, next one is novel, next one is atypical. First one depends on diacylglycerol, calcium, and 4-ball esters. Second one, diacylglycerol and 4-ball esters. Third one doesn't depend on any of these, but some adapter proteins are responsible for activation. That is atypical PKCs. Because it is lacking the usual C1 conserved domain, rather it has got C1-like domain with a single cysteine rich, which cannot bind with this DAG and 4-ball ester. That's why this atypical PKCs require some other cofactors for its activation. Now, the C2 domain of classical PKC is a module of about 120 amino acids that binds phospholipids in calcium-dependent manner, resulting in membrane association of PKCs. So, C2 domain are also found in proteins other than PKCs like phospholipase. So, now C2 domain. So, this is the C2 domain you have in classical PKCs, and this is another C2 domain you have in novel PKC types. Whereas in the atypical PKC, there is no C2 domain. That is about the structure. So this is the way you have to practice the structure, and mainly it is, regulatory domain as we discussed. Novel, classical, they are having more resemblance, but their activators are different. So C1 domain and C2 domain, that towards N terminal, whereas towards the C terminal there is a kinase domain, activation catalytic domain. Okay, so this is PKC regulated related kinase. There is other type of kinase also here written, and you have here cysteine rich. These are cysteine rich sequences, and you have plectrin homology domain. Any questions till then? Any clarifications you require? So for you, first time it may be difficult, but you need to practice and try to understand. Okay. So next one is the signaling cascade, protein kinase C mediated signaling cascades, functions, and its regulation. So today we are going to discuss about the functions. Regulation is a small topic. Next class I will discuss, and that is going to be the last topic of this chapter. So protein kinase C now here, this is the protein kinase C is at the central part of this signaling cascade. So this PKC is getting activated when it is bound to calcium, DAG, and TPM for esters. So this PKC, the phosphorylated form, active form, is basically getting activated in different ways. First way, if you see here, is the receptor tyrosine kinase, activated receptors, which is bound with the ligand. And this is your serpentine receptors, G protein coupled receptors. So both of these are responsible for activation of PKC mediated by another enzyme known as phospholipase C, beta gamma forms. So this beta gamma, one is responsible for beta and another one for gamma. So this way, PLC beta gamma is getting activated by these receptor tyrosine kinases and G protein coupled receptors upon its activation. This in turn responsible for the cellular calcium levels and diacylglycerol, which then binds to PKC. This is mediated by PLC. So that way, PKC is getting activated. Directly, this TPA is very important because this is responsible for controlling the cell proliferation. So this formyl ester TPA, the product of fatty acid metabolism, this can also directly bind to PKC. And in other way, through RAS signaling. So there are some proteins known as RAC. So RAC proteins are the parts of this RAS signaling, which is activated by this growth factor receptors, like epidermal growth factor receptor here, upon binding the ligand. So what is the ligand here? Epidermal growth factor. When it binds to growth factor receptor, this phosphorylated is activated through RAC. So PKC is getting activated through this pathway, that is RAS pathway. This growth factor receptor mediated pathways are RAS pathway, that's MAP kinase pathways, and in this mediated by RAC proteins. These are membrane associated proteins, RAC proteins we are going to discuss next. These RAC proteins are responsible for activation of this PKC in another way. So this way also PKC is getting activated. And another proteins, MRACs, which are also responsible for activation of this PKC. And this PKC also phosphorylate this MRAC. This is the effector. MRAC is effector. Okay. So this PKC, after its activation in different ways, is then responsible for further activation of its downstream substrates, which then in that way controls cell cycle, cancer, redox signaling. Senescence means aging, cell aging, then apoptosis. So this signal pathway you have to practice. This is the PKC signal. So what we discuss now, PKC activation occurs in three ways. So like this here, three ways, 1, 2, 3. So receptor tyrosine kinase via PLC gamma, so gamma beta, right? So receptor tyrosine kinase is by gamma form and GPCRs is through beta form. Receptors is gamma form and GPCR is through beta form. So formyl esters, TPA, and the third one is RAC mediated activation through RAS pathway, and this includes another form of phospholipase. See here the depiction is not there. So phospholipase epsilon, this is another type mediated through the RAC. So these three ways this PKC is getting activated. So receptor controlled signal pathway lead to formation of intracellular messenger substances. Ca2 plus is an important cellular second messenger, right? So substances calcium 2 plus and diacylglycerol. That like formyl esters activate all these three together upon receptor tyrosine kinase and G protein coupled receptor activation, responsible for activation of PKCs. Then PLC beta gamma are the important activators downstream of this activated receptor tyrosine kinase, as the depiction we discussed. PLC beta gamma. So gamma is receptor tyrosine kinase and beta is through G protein coupled receptor activated signaling. So this further responsible for PKC activation through calcium and diacylglycerol. Receptor controlled signaling pathway lead to formation of the intracellular messenger substances. So this way these pathways are responsible for calcium and diacylglycerol, and four esters that together responsible for this is the same point here. Okay, so you can write at the last one instead of here. So this responsible for together activation of PKCs. So phospholipase beta gamma are the important activators. This we discussed, and this is responsible for the activation of PKC through calcium and diacylglycerol. Translocation to the cell membrane is linked with the activation of protein kinase C. Receptors of PKC involve RAC protein. So another pathway is through RAC. What is RAC here? Receptor for activated protein kinase C. So these RAC proteins through the RAS pathway also control this PKC activation. So substrate of protein kinase C is substrates. Okay, that is downstream of protein kinase C. MRACs. MRAC means myristoylated alanine-rich protein C kinase substrate. So MRACs are the substrates. They are not responsible for activation like RAC, but instead, after protein kinase C activation, next one is MRAC. Signal transduction, everything is goes like an arrows. Okay, so this is the theme you need to understand clearly. Which one is responsible for activation of which one? Which one is upstream signal? Which one is downstream? That is all about signal transduction. So proteins and other the MRAC proteins and other proteins associated with the cytoskeleton. These are all the substrates of protein kinase C. Understood? Substrate activated protein kinase C substrates. Once protein kinase C is activated, it is responsible for phosphorylation of MRAC. But MRAC is not doing here. But here is through the RAS pathway. This RAC is responsible for activation of PKC, and that adds to the pool of activated protein kinase. Understood this point? Any questions you have? So even vitamin D3 also is linked with this protein kinase C activation. So that is all for today's discussion, and with this your protein kinase C discussion also over. Regulation small topic is there. So tomorrow or next class, along with this, I will start another topic. So any questions you have, please ask me. Clarifications, doubts, further explanation if you need, you can ask me. Definitely, I can say that there are so many new terminology which you have not come across till now. So you need to practice. It's very important for biochemistry students. All these proteins, names, then pathways, you have to take papers and keep writing. So I'm going to give an assignment where you have to make the pathways of all this for this signal transduction topic. Any questions? All of you understood? Please unmute everyone and tell that you have understood.
Yes, sir.
Yes, sir.
Yes, sir.
Yes, sir.
Yes, sir.
Okay, very good. If you have understood, fine. It is easy, but only thing is there are new terminologies which you have not exposed so far and you are coming across for the first time. So you need to practice, keep memorizing. As I said, take a rough book and keep practicing all these pathways. It is easy because straight questions that you can expect, and if you are well prepared in these things, easily you can score the marks. Okay, then I am stopping here, and I'll continue with the same paper in the next class. See you, everyone.